attached both to St Andrews and James Hutton Institute. Adam wanted me to describe this as a now for something completely different talk, but actually I'm not quite so sure after the last
Not anymore,
talk.
no.
There may be some commonality in between your research on the influence of sediment characteristics on the transport of pathogens from freshwater to coast. Over to you.
Thank you. So yes, I'm a second year PhD student at St Andrews University and the James Hutton Institute in Aberdeen, part of the Sediment Ecology Research Group at St Andrews. Supervisors are Professor David Patterson, Emma Defew at St Andrews, and Lisa Avery and Mark Stutter in Aberdeen. So why am I doing this? So current water quality monitoring for bathing waters is based upon the detection of fecal indicator organisms detected in samples from the water column. Fecal indicator organism is an organism which is relatively easy to detect and culture from a water sample that is not necessarily pathogenic in itself, but as it indicates recent fecal pollution, the more there is, the more chance of actual human pathogens being in the water. So the sediments underlying the water can contain over 1,000 times the abundance of fecal indicator organisms than the overlying water. And obviously when these are resuspended through anthropogenic or natural causes, these will cause adverse water quality at that site or downstream. And by investigating the relationships between fecal indicator organisms in the sediment, so the characteristics of the sediment, so looking at why they are there, and then the erosion risk of how likely is the sediment to move on to bathing waters, and then environmental factors such as rainfall and wind direction, tides, we can look at preconditions to the adverse water quality that stops people going to bathing waters. So yeah, the current water monitoring by SEPA doesn't take into account fecal indicator organisms in the sediments. So it doesn't account for the bather going into the water and stirring up any sediment. They have to swim in and swim out. And also it doesn't account for beachgoers who don't go into the water, because obviously the water will flood up the beach and pathogens in the water will settle out into the sediment. The water will go away, people will visit there. The other issue is that water quality monitoring only occurs during the bathing season, which might be quite long in the south of England, but up in Aberdeen it's about 2 or 3 days on a bank holiday in June. So the other issue with that is, as you'll see later, the E. coli counts tend to be higher during the winter rather than the summer. And so people dog walking in the winter there's no risk assessment for them. So the bathing water regulations state that SEPA should publish where a certain bathing water is likely to be affected by short-term pollution and publish the conditions likely to cause this short-term pollution, which is obviously useful for the public when they're going to a beach. And a lot of places have electronic signs. But how do they know what the risk is to the public if they don't know what's going to be resuspended from where, from what sediments at what time? If they know there's sort of industrial work happening upriver or up the coast, suspended sediment might be coming down, and then that might settle out for a few weeks and then get resuspended at a later time and cause problems then. So that's kind of the point of the project, is to develop a tool or to kind of identify what sediments contain the high abundance of fecal indicator organisms at what time of the year, and how likely these will be to resuspend to high-risk areas like bathing waters. So Lundiston Bay is the closest monitored bathing water site to Glasgow, just opposite Dunoon, so slightly out of the inner estuary, but it was designated in 2008, and since then it's only had 2 years where it's achieved a guideline pass, which is under 100 E. coli colony-forming units. So E. coli and enterococci are the 2 common fecal indicator organisms. But the mandatory standard is under 2,000 E. coli per 100 ml, which, put into context, 10 cells of enterhemorrhagic E. coli 157 is enough to give you a very bad poisoning. So that is 0.5 ml of water, and that's when you've swum into the water and not stirred up any sediment, which contains 1,000 times more than the water itself. So they detected a peak of 1,600 E. coli per 100 ml in July 2013, and my data shows from the East Coast that I'm looking at the moment, it's worse in winter. So this is likely to be higher in winter as well. So my fieldwork is the Hutton estuary, 20 miles north of Aberdeen. It's got relatively low annual rainfall, summer temperatures of 14 degrees and winter of 3 degrees. The catchment supports 50,000 wildfowl and 1,000 seals reside at the estuary mouth, and the estuary is only 7 kilometres, so there's quite a source of fecal contamination from lots of warm-blooded animals. So I have 4 monthly sampling sites. The upper reach, which is a lot lower salinity, and it's kind of a mixed mud. Then I have the mid reaches where there's one out on a sand spit, so it's very high percentage of coarse grains, and then set back in the bay where there's very high percent of organic content and fine sediment settling out. And then the lower reach, which is again a mixed sand, so sand with some muds. The analysis I'm doing, so we've got the coliforms and E. coli. Coliforms are another indicator of water sanitary, the sanitary condition of the water. This includes kind of a wider range of bacteria such as Enterobacters, Citrobacters, and Klebsiellas. And we've got a technique where you put in a supernatant from a sediment sample and heat seal it into separate wells, and there's 2 pigment-labeled nutrients which, when broken down, turn the well to a yellow colour, as you can see, when there's coliforms present, and E. coli make the wells fluoresce under UV light. So we're also looking at the erosion susceptibility of the sediment. So that's the CSM that Kate mentioned earlier, which is a water-filled chamber on top of the sediment surface, which you measure the transmission across of light, and increasing jets of water hit the sediment surface, and you look at the transmission as sediment is kicked up off the surface by increasing increasing powers of jets of water. And that's to kind of look at the fine-scale surface erosion. And then there is the shear vein, which is mass erosion of the bed. So we're also looking at the bulk density, the water content, the organic content, and then the chemistry of the interstitial water, sort of the redox, the salinity, the pH, and a host of other analyses, sort of carbohydrate, protein, grain size, surface area, and environmental factors. So it's the first data from the monthly sites. It's kind of arranged 1, 2, 3, 4 from sandiest to muddiest. But these labels are hard to use because it's constantly getting eroded and deposited in the estuary, so it never stays sandy or mixed sand. It kind of chops and changes, which isn't good for diagrams. But you can see in all the graphs there's a trend of decreasing colony-forming units of E. coli with depth. This is probably because the chemistry of 5 centimetres down is a lot more hostile to an organism which is used to living in 37 degrees in a homeotherm gut. And then the 2 lower ones here with the mud don't have quite as much of a decrease. This is possibly due to there's more biota in the mud, sort of turning over the sediment with bioturbation, forming not quite a hostile environment deeper down. Okay, so for the last 6 months, this is each of the sites. As you can see, there is a downward trend as it gets warmer. And this is the problem with using the sand mix sand as labels, because such as in March, we got deposition on the mixed sand, which was higher in organic content, because in March all the sites decreased in E. coli numbers, apart from one which had organic matter deposited, so E. coli survived better. That's one problem with displaying the data like this, so this is why we're going to be using sort of a modelling approach to it, using the variables separated from the times of year, and just using temperature and rainfall as kind of the environmental factors. Okay, so we're also doing a quarterly transact of the Eden Estuary in St Andrews and the Ithen Estuary in Aberdeen. So the E. coli concentrations in the sediment are higher the higher up the watercourse you go, and they get diluted, and there's larger grain size, so less surfaces to absorb to, closer to the mouth. But you can see on both estuaries there's kind of a corner. Both estuaries sort of 4 or 5 points down, and this, because it's a corner, it's a high flow, and there's slightly larger grain size deposited, and these have quite a lot less E. coli in the sediments compared to the bays afterwards. And this is some of the results from that same quarterly sampling in February. So the top two graphs is salinity against E. coli, and you can see as salinity rises, E. coli decreases. But especially on the Eden, you can see it's not a straight line. It's not the easiest pattern, so there's plenty other variables to look at and model effectively. The table at the bottom is some quick linear regressions from some of the variables paired with the E. coli. You can see, so water content and organic content are the main drivers of E. coli distribution and abundance in the estuaries. And as Kate said before, the cohesive strength is a complicated issue. So there's no correlations with it, which is worrying in a way, because there's no way of knowing at this stage which sediments are likely to resuspend. There's no correlation between those which have the most E. coli in and the ones which are likely to be moving and contaminating bathing waters. So there's no link yet to know which are the most high-risk sediments. So early conclusions are the depth profiles, that E. coli and coliform abundance decreases with depth across all the sediment types examined. The finer sediments allowing the indicator organisms to persist for longer at a depth. And the long-term monitoring results, well, in our early stages, there's some correlation with E. coli with several variables, but it's to be modelled with RAML when we've got the full data set. I'm quite confident that we should get some very interesting results, especially down to the temperature, because as you'd expect more contamination in warmer weather. So we're beginning to gain an understanding of the main drivers of E. coli abundance. And so this is to inform policymakers with what we found and what sites maybe need more monitoring of sediments. If, say, coastal management, if you change your coastal management further up the Clyde, you'll change the hydrology of the estuary, and you'll change what sediments are getting washed away from where, and you'll get settling out of different sediments in different areas that you expect, and you don't know the consequences of this to the E. coli distribution. So other work I'm doing is I'm doing molecular work, terminal restriction fragment length polymorphism, which is taking genes from a set group of organisms, such as algae, and then looking for single or small morphs in their DNA to characterize the biodiversity. So we're doing that on algae, cyanobacteria, bacteria, and looking at how this affects looking at how this changes with the different sediment types, but also how it affects E. coli populations because when it's warmer, the reason that possibly when it's warmer the E. coli drops is because the native biota is more active, so you get prokaryotes grazing, which eat bacteria, and an E. coli is out of its native environment. It's defenseless compared to other bacteria in the sediment, and also because of that it can't compete as well against native bacteria. We're also looking at the viable but non-culturable state of E. coli using quantitative PCR, which is counting DNA instead of whether the bacteria can be cultured or not, because E. coli enter a state where they kind of go dormant, almost hibernate until they are back into homeotherm gut, so they're undetectable. And we're also doing a bit of infrared spectroscopy to characterize the sediments. So a couple of minutes you can get a spectra like that. This is just a portion, about a tenth of the whole spectrum, and from there you can look at two forms of calcium carbonate, and you've got a clay in there as well. And if you work hard enough, you can start to kind of semi-quantify how much of each you've got in each of your samples. So, thank you.
Questions from the audience? Cecile first.
I was wondering, did you choose the sample size and if you managed to compare similar geologies along the river?
So similar geologies along the river, that's kind of what the quarterly sampling is for. So the monthly is picking 4 different geologies. The quarterly is trying to incorporate as many different types as we can. And because it's constantly changing, along with the monthly sites are constantly changing with their mud and sand fractions. So instead of having 4 distinct, we are kind of getting a broader range spectrum, as instead of 4 pinpoints, there is quite a range of the sediments. And that was also the point of doing the Eden estuary and the Ithen estuary. You'd have different types of clay washing down because of the geology of the areas.
Just wondering why there was a negative correlation between E. coli and salinity. Is it just a dilution thing? That you're getting more seawater, so you're getting dilution?
That's one thing. It could be, but also we found that in some of the sediments where it's on a hot day in the last couple of months, where a sediment has been exposed to sun and it reaches sort of 22 degrees, you get a lot of evaporation of the water, so you create a hypersaline environment. I think, again, that's it's not native. The E. coli is not native to the estuary, and a hypersaline environment will kill it off or make it adopt this dormant state.